Hi everyone, today we're presenting our project on designer babies. Our group members are me, Carmen Goodman, Samantha, Sydney, and Wallace. In this presentation, we're going to explore what designer babies are and how gene editing works, and also the scientific, ethical, and social issues surrounding this technology. We'll also look into how this process connects to IVF and the risks involved, and whether or not society should allow genetic modifications in humans. So let's get into it. Let's start our summit of ethics. So first, what exactly is a designer baby? Well, that's an important question. A designer baby is a genetically modified embryo that was altered using gene editing technology, most commonly known as CRISPR-Cas9. These embryos form designer babies. The process mostly used to create designer babies is known as IVF. To fully understand what a designer baby is, we need to understand what IVF is. For those that don't know, IVF stands for in vitro fertilization, and this means the fertilization happens outside the body, usually in a lab. This process is commonly used for people who have trouble getting pregnant, including women with conditions like endometriosis, ovarian disorders, or blocked fallopian tubes. It is also used for couples with genetic conditions, same-sex couples, in cases of male infertility. IVF makes it possible to create embryos that can later be screened and edited before implantation. The IVF process starts with hormone injections that stimulate the ovaries to produce multiple eggs instead of just one. When the eggs are ready, they are collected and fertilized with sperm in the lab. If the sperm is weak, a technique called intracytoplasmic sperm injection, or ICSI, can be used to inject a single sperm directly into the egg. After the fertilization, the embryos are allowed to develop for a few days, and then one or more are transferred into the uterus. One risk of IVF is multiple birth, since more than one embryo can be implanted. Now that we have this background knowledge, let's get back to how gene editing actually works. While using the method CRISPR-Cas9, IVF is used during the in vitro fertilization part. While the sperm is being injected into the egg, they can genetically modify the embryo before implantation. They do this by identifying specific DNA sequence they want to change, and they can design something called a guide RNA, which directs the Casper system to the exact location in the genome. By doing this, the CRISPR-Cas9 complex binds to the DNA and cuts into a precise location. And once the DNA is cut, the cell repairs itself, and during this process, scientists can insert, remove, or modify genetic material. This allows scientists to potentially remove harmful mutations or even introduce new traits into the embryo. While this technology is a marvel and revolutionary, it is not an exact science. It has not been perfected. In the next coming slides, we'll look into the current limitations and risk of gene editing.
IVF incorporating CRISPR-Cas9. During in vitro fertilization, or IVF for short, embryos develop into in vitro, which refers to experimentation outside of the body. CRISPR-Cas9, in this case, can be used at this stage to remove harmful genetic mutations and potentially select desired traits. It's currently used mainly for research purposes, with the focus on correcting inherited genetic diseases. The role of OCT4 in development. The PAL5F1 gene produces OCT4 protein, which is a key transcription factor regulating gene expressions and helps maintain stem cell self-renewal and pluripotency, which is essential for early embryonic development, and acts as a marker of stemness, also seen in some cancer cells. Why this matters? Gene editing targets critical development genes like OCT4. Small changes can have major developmental effects. It highlights both medical potential and ethical and biological risk. Now, how does gene editing technology work? Well, first things first, you have to identify and target the DNA sequence, then design the single guide RNA, and then form the CRISPR-Cas9 complex, then locate the PAM and target DNA, cleave the DNA, and then repair the DNA. CRISPR-Cas9 mechanism. CRISPR-Cas9 is a tool used to precisely modify DNA sequences. It uses guide RNA or gRNA to locate a specific gene in the genome. Cas9 enzyme acts as molecular scissors to cut DNA at that exact spot that's needed to be cut. The cuts create a double strand break, triggering the cell's natural repair system. Now there are two repair pathways for this. NHEJ, or non-homologous end joining. It's a quick but error-prone repair pathway that inserts or deletes DNA, which can turn off the genes. Then there's HDR, or homology-directed repair, which is a more precise repair pathway that uses a template to accurately fix or replace genes. The possible outcomes of this is the removal of harmful mutations, insertion of new genetic material, or modifying gene expression. CRISPR-Cas9 can be applied to treating genetic diseases, researching genetic functions, and potentially using it in embryos during in vitro fertilization. The limitations of it are off-target edits or wrong DNA cuts, unintended mutations, and ethical concerns and unknown long-term effects.
If we have time, we can play the video at a later moment, but we're going to proceed with the slides. Wallace, can you take over from here?
Now let's go back to current limitations on CRISPR-Cas9 again. Like I mentioned before, off-target effects, precision issues in editing can cause unintended mutations. Efficiency and repair mechanisms. Efficiency issues with editing does not guarantee proper results. Another one is delivery challenges. Practical issues arise due to the adeno-associated virus, or AAV, having a small capacity in the protein from Streptococcus pyogenes. Sorry if I'm messing this up. Pyogenes, or SBCase9 is too large. Researchers are forced to use smaller AAV. However, it can potentially cause second cognitive vectors. Tissue-specific targeting. The CRISPR components can be delivered to the desired organs, but in doing so, it can affect their tissues. Immune response. CRISPR-Cas9 proteins are derived from bacteria attacking the edited cells that have T cells or antibodies. And last but not least, ethical and regulatory limitations. Editing human genomes can create ethical risks and dilemmas if not regulated properly.
Hello everyone, my name is Samantha and I will be continuing the presentation of the seminar on ethics. A big ethical question is, should we even be editing genes? On one side, gene editing could be used to prevent serious diseases like cystic fibrosis or Huntington's disease, and this could save lives and reduce suffering. But it also raises the question, where do we draw the line between treatment and enhancement? For example, should parents be allowed to choose the traits of their children like intelligence, appearance, height, or athletic ability. And if that becomes normal, it could create pressure on children to meet these expectations and change how we define what's normal or ideal. Another major issue is access, specifically who gets access. Gene editing and IVF procedures are very expensive, so not just anyone can be able to afford them. This could create a divide where only wealthier people have access to genetic enhancements and disease prevention. And over time, this could create social inequality where certain groups have biological advantages over others. This could lead to discrimination. If some people are genetically edited while other people are not, society may begin to value certain traits more than others. This leads to people without genetic editing to be judged or seen as less desirable, and it can create a new form of genetic discrimination where opportunities are influenced by someone's genetic traits rather than their abilities or character. One major concern is the idea of a genetic advantage. People who can afford genetic editing could gain built-in advantages, creating a divide between enhanced and non-enhanced individuals. This could reinforce existing biases related to things like race, gender, intelligence, or physical traits. There's also the risk of stigma. People who aren't edited may be judged, while edited individuals may feel pressure to perform. Without strong regulations, this could even lead to discrimination in jobs, education, and insurance. Overall, it raises a serious ethical concern: the possibility of a modern form of eugenics where certain traits are favored over others. A real-world example is the CRISPR baby experiment conducted by He Jiankui. Scientifically, the experiment had major flaws. It was performed on twins who had mosaic edits, meaning that not all the other cells were edited in the same way, and there were possible unintended mutations. The goal was to make them resistant to HIV by modifying the CCR5 gene, but this may have actually increased the risk for other infections. Also, this wasn't even necessary because HIV transmission could have been prevented by using safer methods like sperm washing. There was also serious ethical and legal violations, including lack of proper consent and illegal germline editing, meaning that the changes could be passed to possible future generations. Globally, this caused major concerns and damaged trust in gene editing research while raising fears about designer babies. Most importantly, there's no clear evidence that the experiment even worked, and the babies were not proven to have gained immunity to HIV.
Now we're on our last section. Take it away, Sydney.
So, what are the long-term risks? These include genome instability, which includes large unintended edits that can destabilize the genome, and instability can also disrupt normal chromosome structure and then increase the chances of mutation. Another long-term risk are the disruptions of essential genes in regulatory regions, which are deletion or altered nearby genes, which are not intended for editing, and it also interferes gene regulatory like promoters and enhancers, and also includes abnormal gene expression. Another long-term risk is also mosaicism, which includes not all cells being edited the same, which then can lead to uneven tissue function and harmful cell pop. So what unintended mutations can occur? In an individual who undergoes gene editing can lead to somatic consequences, such as altered cell behavior. It could also lead to delayed effects, such as silent mutation that don't activate till later in life. And it also leads to clonal expansion, such as cells with advantageous mutation that can grow over time. So who will regulate and enforce this technology? Historically, when governments try to control aspects of a person's agency and medical choices, the voiceless tend to suffer the most. Examples of this are from the Milgram experiment, which was an obedience of authority test that shows how easily people can be pressured into bad choices if an important figure was backing the push in direction. Also, the Ask conformity experiment shows the dangers of society pressure, and people in the experiment would change their answers to the correct in fear of being an outlier. And this finally leads to the question, are we playing God or extending medical progress?
What kind of future do you want? Does anyone have any questions?
Hello.